All right, so let's pick up where we left off with chapter 13 with sensations, part 2, the role of the peripheral nervous system in sensation. So, stimuli that are first detected by sensory neurons get transmitted by sensory neurons to the central nervous system, where that stimulus is integrated and interpreted by the central nervous system neurons, and then appropriate action will take place. Sensory transduction is when stimulus is converted into an electrical signal. The ion channels in the axolemma are closed. The stimulus is detected by the sensory receptor. The sodium ion channels open. Sodium ions then flow into the axoplasm, causing a temporary depolarization, which we refer to as receptor potential. If enough sodium ion enters, then the membrane potential may reach its threshold. Once that has occurred, the voltage-gated sodium ion channels will open. The action potential has been propagated along the axon toward the central nervous system. So here's a probe poking the finger. The somatic sensory nerve, it's either the unipolar neuron, sends the signal. The sodium channels will open. The sodium will begin to flow through, and if enough reaches for the potential to occur, then the reaction will take place. So here we see the probe now depressing the skin, causing the pressure. Those sodium channels are opening. Sodium is flowing through. Then once the action potential is reached, the threshold is reached, then the action potential moves to the next neuron. Sensory transduction rapidly adapts these receptors. They respond rapidly and with high intensity to stimuli. They will stop sending signals after a certain time period called adaptation. The receptors will then detect initiation of stimuli, but ignore ongoing stimuli. That's kind of the thing where you think about a smell that you smell, and then it's very intense, but it begins to weaken over time, and then over an extended period of time, you no longer detect it. You see this in places with usually unpleasant odors. You go into an area and you go, oh my God, what does that smell? How can y'all possibly stand it? And people who have been in the room for 30 minutes say they don't even smell it anymore. Slowly adapting receptors respond to stimuli with constant action potentials that don't diminish over time. So there are certain receptors that you will always detect whatever sensory input that is being detected. Sensory receptors exist in many forms. You have encapsulated nerve endings, which are surrounded by specialized support cells. You have free nerve endings, which don't have any support. You have exteroceptors, which are usually close to the body's surface, as their name would imply, external. They detect stimuli originating from outside the body. Interoceptors are found within the body's interior. They detect things originating from inside the body. You have mechanoreceptors, which are encapsulated interoceptors or exteroceptors in the musculoskeletal system, the skin, and many other of the organs. These depolarize in response to anything that mechanically deforms the tissue where the receptors are found. That mechanical deformation causes those mechanically gated ion channels to open, which allows the sensory transduction signal to go forward. So things like vibration, light touch, stretch, and pressure are examples of mechanoreceptors. Thermoreceptors, as their name implies, respond to temperature changes. They are exteroceptors. They are mostly slowly adapting receptors. And there are separate receptors that detect hot and cold. So when you feel it's hot, it's one set of receptors. When you feel something is cold, it's a different set of receptors. The chemoreceptors can be either intero or exteroceptors. They depolarize in response to binding with specific chemicals, either in body fluids or in the air. and they generate a receptor potential as the sodium ion channels open. Photoreceptors are found only in the eye. They depolarize in response to light, and nociceptors are usually slowly adapting exteroceptors. They depolarize in response to noxious stimuli. So noxious, think of unpleasant. So nociceptors are those things that detect pain. Capsaicin, that's the product in hot peppers that makes you feel like your mouth's on fire. It opens very specific ligand-gated channels in the nociceptors, triggers action potentials, causes the central nervous system chemical, or is perceived as painful. Repeated application of capsaicin to nociceptors seems to desensitize them, makes them less likely to generate receptor potentials in response to the painful stimuli. Therefore, capsaicin is applied as a topical cream to relieve pain in peripheral nerve disorders called neuropathies, or shingles is another example, and some other conditions. If you have a topical pain cream, if you look over at the back of the ingredients, you will probably see some level of capsaicin in there. Doesn't treat the actual cause of the pain. It simply makes the nociceptors less able to relay pain stimuli to the central nervous system. Sensoroceptors, the mechanoceptors, you have Merkel cell fibers. These are slow adapting nerve fibers surrounded by a capsule of Merkel cells. These are found in the epidermal ridges of the integumentary systems, primarily in the hands, especially in the fingertips. These receptor potentials are generated by mechanically gated ion channels. These are the things that detect discriminative touch stimuli. You know, what is an object's form? What is its texture? Is it smooth? Is it rough? Is it round? Is it square? Does it have corners? Does it not? All of those things are found with your Merkel fibers. Meissner's corpuscles, or your tactile corpuscles, are found in the dermal papillae. They are rapidly adapting tactile exteroceptors. They discriminate touch stimuli. The Ruffini endings, or the bulbous corpuscles, are spindle-shaped receptors in the dermis, hypodermis, and in the ligaments. Again, they are slowly adapting receptors that respond to stretch and movement. Laminated corpuscles, or Pacinian corpuscles, these are layered. When you look at one, they sort of look like an onion that's been sliced in half in cross section. They rapidly adapt these are rapidly adapting receptors found deep within the dermis. They detect high frequency vibration and deep pressure stimuli. When you looked at the skin model, or when you do look at the skin model in lab, you will see these Pacinian corpuscles. Hair follicle receptors are free nerve endings around the base of each hair, found in thin skin. Hair follicles are not found in the soles of the feet and the palms of the hand. These respond to stimuli that cause the hair to bend. Proprioceptors detect movement in position or joint body parts. These are found in the skeletal muscle system. And there again, cross section of the human integumentary system. There's the epidermis, there's the dermis. This area down here is referred to as the hypodermis. There's the laminated corpuscle, there's the Merkel cells, tactile corpuscles, Ruffini's endings, and the laminated, that's the one that we call the Pacinian corpuscle. Types of thermoreceptors. You have cold receptors that respond to temperatures between 10 degrees Celsius and 40 degrees Celsius. Again, that's 50 to 104. These are found in the superficial part of the dermis. Hot receptors, again, temperatures between 32 and 48. These are found deep in the dermis. The temperature outside these ranges are detected by nociceptors. That's the reason extremes of temperature, either hot or cold, are interpreted as pain. These temperatures are the ones where we're just reacting to the temperature of the ambient room or environmental temperature. If it gets above 20 or below 40, it starts to be uncomfortable. Those temperatures are nociceptors. Sensory neurons, the first order somatic sensory neurons are pseudo unipolar neurons with three main components. There's a cell body. The cell body of spinal nerves are in the posterior root ganglion, just lateral to the spinal cord. The cell bodies of the cranial nerves are in the cranial nerve ganglia in the head and neck. The peripheral process is a long axon that transmits action potentials from the source of the stimuli to the neuron central process. And the central process exits the cell body and travels through the posterior root, enters the spinal cord at the posterior horn or the brainstem if you're talking about cranial nerves, where they deliver their action potentials. Peripheral process transmits action potentials from the sensory neuron receptor to the neuron central process. The central process then transmits the action potential from the peripheral process to the posterior horn, eventually synapsing on the second order neuron in the spinal cord or the brainstem. The action potential gets propagated down the peripheral process, doesn't generally reach the cell body. Instead, it's transmitted to the central process in the area where the peripheral and central processes come together in contact near the cell body. I was hoping there would be a picture of these in just a second. So here's the neuron, here's the cell body, there's the central process of the somatic sensory nerve, and there's peripheral process. You see it's running all the way out to the nerve endings. Signal comes in, and then we'll go to the, and generally they're going to bypass this nerve. They're just going to cross directly across and go into the posterior horn of the spinal cord. Sensory neurons are classified by two factors that determine speed, the speed at which they produce their action potentials, the diameter of the axon, and the thickness of the myelin sheath. So large diameter axons with thick sheaths conduct the fastest impulses. Proprioceptors sending information to the central nervous system, or that discriminative and non-discriminative touch information. The smaller diameters with little myelin transmit action potential slowly. They include axons that carry pain and temperature stimuli to the central nervous system. Receptive fields are the areas served by a particular neuron. Neurons with more branches innervate larger receptive fields. Body regions whose primary functions is sensing the environment, such as our fingertips, contain many neurons with small receptive fields. Those body regions that are not as involved in sensing the environment, again, the skin of your forearm, for instance, have fewer neurons with larger receptive fields. A two-point discrimination threshold is a method for measuring the relative size of receptive fields. So receptive fields, fingertips, see these receptive fields cover quite a bit of surface area. Then you have areas here. Two-point discrimination, threshold of about 3 millimeters. In other words, if you have probes and you're poking over 3 millimeters apart, you would experience that as as two sensations. But down here, for there to be two sensations, you have to be about 40 millimeters apart. So if you're just 1 or 2 millimeters apart with a probe, and you've got two probes, and you're poking somebody on the finger, say 1.5 millimeters apart on your fingertips, your body is going to discern that as one signal. Down here, you have to get 40 millimeters apart before your body starts to recognize that there are two stimuli there. Durotomes are, the skin is divided into different segments, or dermatomes, based on the spinal nerve that supplies the region with somatic sensation. Dermatomes can be combined to assemble a dermatome map, which represents all of the sensory pathways to the different parts of the body, except for the first cranial nerve, first cervical spinal nerve. And dermatome maps can be used clinically to test in the integrity of sensory pathways to different parts of the body. And again, here's where an example of a dermatome map, and somebody doesn't have any sensation in this area of the body for some reason, then they have a problem with their L2 spinal nerve. If something's going on in this part of the head, numbness or something, then there is an issue with the C2 nerve. Referred pain is a phenomenon whereby pain that originates in an organ is perceived as cutaneous pain. This occurs because many of the spinal nerves carry both somatic and visceral neurons. So when the visceral sensation travels along the same pathways as the somatic sensations, therefore the body is recognizing the pain as being a dermal and not a visceral pain. Referred pain is generally located along the dermatome for a particular nerve. Again, see, here's the map for the heart, where the area where the heart is. Here's some other areas of the heart. So when someone has a heart attack, they have pain in their chest, but it also runs down their left hand or arm and can be in their throat or in the middle point of their back. Liver and gallbladder problems, you can have issues in your shoulder, the middle of your back, or toward the bottom. Kidneys will be along your back, but will radiate down on either side. Here again, you see that radiating down. Bladder issues, but can also play a role in the small intestine. There's the colon problems, ovaries in women. All of these areas that are the same color, you may be seeing some similar you're feeling it in your skin, but the pain is actually originating somewhere else. So the big picture of detection and interpretation of somatic sensation by the nervous system. Big picture, signal comes in for the peripheral process, the action potential moves down, crosses this sort of bridge here by the cell body, moves into the proximal process, or the central process, excuse me, where it moves into the lateral horn of the spinal cord, crosses over, because remember, pain on the right side is affected on the left, crosses over and moves up to the third order neuron, where we are finally told that there is pain. And then the signal goes back if an action is required. Okay. Let's stop there, and we will pick up again after a while.